Hello. This is the Unit 3, Module 10, Chapter 20C presentation. The big picture for this Chapter 20, Part C presentation includes the mechanisms of action of antiviral drugs, a discussion of the mechanisms of action of antifungal drugs, and a very brief mention of the drugs used against parasites. Compared with the number of drugs available to treat bacterial infections, the number of antiviral drugs is very small. The major reason for this difference is the difficulty in obtaining selective toxicity against viruses. Their replication is intimately involved with the normal synthetic processes of the cell. Another limitation of antiviral drugs is that they are relatively ineffective because many cycles of viral replication occur during the incubation period when the patient is well. By the time the patient has a recognizable systemic viral disease, the virus has spread throughout the body and it is too late to interdict it. Furthermore, some viruses, for example herpes virus, become latent within cells and no current antiviral drug can eradicate them. Table 20.2 summarizes the characteristics of antiviral drugs. Enfuviritide, also known as Fusion, is a synthetic peptide that binds to the protein GP41 on the surface of human immunodeficiency virus. Fusion thereby blocks the entry of the virus into the cell. It is the first of a new class of anti-HIV drugs known as fusion inhibitors. In other words, they prevent the fusion of the viral envelope of HIV with the cell membrane. Amantadine blocks the replication of influenza A virus. It prevents replication because it inhibits the uncoating of the virus. Other antivirals include nucleoside inhibitor drugs that are analogs of nucleosides that inhibit the DNA polymerase of one or more members of the herpes virus family. The drug acyclovir inhibits the DNA polymerase of herpes simplex virus types 1 and 2, that's HSV 1 and 2, and varicella zoster virus, but it does not affect cytomegalovirus. Acyclovir is a guanosine analog. Ganciclovir is a nucleoside analog of guanosine as well, with a 4-carbon fragment in place of the normal sugar ribose. It is structurally similar to acyclovir, but it has activity against cytomegalovirus. The selective toxicity of nucleoside reverse transcriptase inhibitors, or NRTIs, is based on their ability to inhibit DNA synthesis by the reverse transcriptase of HIV to a much greater extent than they inhibit DNA polymerase synthesis by that polymerase in human cells. Nucleoside inhibitors inhibit the DNA polymerase of herpes viruses by mechanisms distinct from the nucleoside analogs previously described. Foscarnet is the only approved drug in the class at this time. The non-nucleoside reverse transcriptase inhibitors, the NNRTIs, act by binding near the active site of the reverse transcriptase and inducing a conformational change that inhibits the synthesis of viral DNA. Members of the protease inhibitor class of antivirals inhibit the protease that is encoded by HIV. The protease enzyme cleaves the GAG and POL precursor polypeptides which produce several nucleocapsid proteins of HIV, as well as some enzymatic proteins like reverse transcriptase that are required for viral replication. Raltegravir is an integrase inhibitor that blocks the HIV-encoded integrase that mediates the integration of the newly synthesized viral DNA into the host DNA. Tamiflu and its relatives inhibit the neuraminidase of influenza virus. Neuraminidase enzyme is located on the surface of influenza virus and is required for the release of the virus from infected cells. Inhibition of release of influenza virus limits the infection by reducing the spread of virus from one cell to another. Now let's discuss drugs used to treat fungal infections. This slide summarizes in cartoon form the drugs and their specific action on the fungal cell wall. Compared with antibacterial agents, relatively few antimicrobials are available for treatment of fungal infections. Many substances with antifungal activity have proved to be unstable, to be toxic to humans, or to have undesirable pharmacologic characteristics such as poor diffusion into tissues. Fortunately, most fungal infections are self-limiting and require no chemotherapy. Amphotericin B is the most important antifungal drug. It is a polyene used in the treatment of a variety of disseminated fungal diseases. It disrupts the fungal membrane of fungi because of its affinity for ergosterol, a component of fungal membranes, but not a component of bacterial or human cell membranes. Fungi resistant to amphotericin B have rarely been recovered from patient specimens. Azoles are antifungal drugs that act by inhibiting ergosterol synthesis. Some are used to treat systemic fungal diseases. Others are used only topically because they are too toxic to be given systemically. Alkylamines inhibit an enzyme called squalene epoxidase, which is required for the synthesis of ergosterol. Echinocandins are lipopeptides. These block fungal cell wall synthesis by inhibiting the enzyme that synthesizes a compound called glucan, and glucan is not found in human cells. Griseofulvin is an antifungal drug that is useful in the treatment of hair and nail infections caused by dermatophytes. It binds to tubulin in microtubules and acts by preventing formation of the mitotic spindle. Flucytosine is an antifungal drug that inhibits DNA synthesis. It is a nucleoside analog that is metabolized to a compound called fluorouracil. Fluorouracil is the active form, and it inhibits the enzyme thymidylate synthetase. This enzyme thereby limits in the fungal cell the supply of thymidine. Superficial mycoses are often treated, but topical therapy can be used, thus limiting toxicity to the host. The remaining small group of deep mycoses that are uncontrolled by the host's immune system require the prolonged use of antifungal drugs. This, combined with the fact that most of the patients have underlying immunosuppression, makes these infections among the most difficult of all infectious diseases to treat successfully. This table, 20.3, summarizes the antifungal drugs. The study and management of parasitic diseases were seminal to the initiation of chemotherapeutic era. Amazonian Indians were discovered by the Jesuits to be using quinine-containing extracts of cinchona tree bark to treat malarious patients more than 400 years ago. It was in the attempt to synthesize this same anti-malarial compound that 19th century German chemists discovered aniline dyes. The circle closed in the early years of this century when Paul Ehrlich, while investigating the suitability of these dyes as protozoan stains, developed the concept that chemicals might be found to have the capacity to destroy microbial pathogens selectively without damage to the tissues of the host. Although the most dramatic confirmation of that concept came with the introduction of compound 606 for the treatment of syphilis, which we've discussed, Ehrlich's first successful chemotherapeutics were directed against protozoan agents. By 1930, chemically synthesized drugs had been marketed for the treatment of malaria, trypanosomiasis, and schistosomiasis. Few antimicrobials are effective against parasites because they share the eukaryotic characteristics of their hosts. Most of the effective drugs target either neuromuscular targets in the parasites or biochemical pathways within the parasites. Pharmaceutical development of antiparasitic drugs has lagged. Lacking safer alternatives, chemotherapeutics made in the pre-antibiotic era have remained critical components of treatment until recently. Most antiparasitic drugs required prolonged or intravenous administration. Their effectiveness was limited to particular disease stages. Their toxicity mandated that their use be limited to severe or life-threatening infections. Over time, and at a slower pace than antibacterials, newer antiparasitic agents were developed that overcame many of these problems. They are fewer in number, and only recently has their safety and efficacy begun to match those of antibiotics. This slide contains questions to reflect upon and use the presented information. Use the learning objectives for Chapter 20 to guide your study of the information from both the presentation and the textbook. Be sure to define all vocabulary terms. Take the Module 10 vocabulary quiz. When you are ready, take the Chapter 20 test. When you have reviewed the Unit 3 material and you are ready, take the Unit 3 exam. This concludes the Unit 3, Module 10, Chapter 20 presentation.